Bio Photosynthesis Part II is the second of 4 movies that cover photosynthesis. Part 2, somewhat like Part 1, covers photosynthesis in broad terms. This movie provides an overview of the light-dependent and light-independent reactions in photosynthesis. It also looks at the abiotic factors that influence the rate of photosynthesis and gives an introduction to chloroplast structure. The chloroplast, as you know, is the organelle within which photosynthesis occurs. The essential idea is photosynthesis uses the energy in sunlight to produce the chemical energy needed for life. Here is the outline of all the movies available for the photosynthesis unit. Use this outline to find the movie you need for review. Only the first 2 movies are mandatory for SL students, and this movie is focused here. Here is the overall equation, the summary equation for photosynthesis. Study the equation. Get to know the equation well. The transformation of solar energy to chemical energy, as represented by this equation, involves the uptake of carbon dioxide from the air. Water arrives to the plant from the soil through the roots. Solar energy is absorbed by pigments within the leaf, and an organic molecule composed of carbon, called glucose, is the important product. Oxygen is released as a waste molecule. Carbon dioxide diffuses from the atmosphere or from the water into autotrophs. In aquatic environments, the carbon dioxide is available directly from the water. Carbon dioxide is produced by respiration and diffuses out of organisms into the water or the atmosphere. In photosynthesis, the photolysis, the photobreaking of water, produces all of the oxygen released as waste. Thus, 12 water molecules with 12 oxygen atoms are required to produce the 12 oxygen atoms given off as 6 molecules of oxygen. Thus, 12 water molecules, and in turn, to balance the equation, there are 6 water molecules on the right. The glucose formed from photosynthesis represents the chemical energy carbon compound that is available for the energy needs of the plant as well as any consumer that feeds off the plant. Photosynthesis can be measured by carbon dioxide uptake directly, oxygen release directly, or as a change in biomass, which is indirect because as the plant photosynthesizes, it also respires, giving off carbon dioxide. Light shined on an aquatic plant results in the production of oxygen bubbles that can be collected by water displacement. Photosynthesis can be measured directly by the volume of oxygen produced in milliliters per unit area of leaf per time. A plant within a closed container sealed with a carbon dioxide probe can be used to measure photosynthesis. Photosynthesis takes up carbon dioxide, so CO2 levels, concentrations in this case, go down within the chamber while light is shining on the plant. Photosynthesis can be measured directly by the change in the quantity of carbon dioxide taken up in a closed container, parts per million, which is a concentration per area of leaf per time, or could be measured in terms of volume, milliliters of CO2 per unit time. And the rate of photosynthesis can be measured indirectly by the increase in biomass, over time. Measuring mass is an indirect measure of photosynthesis because change in mass is not solely a result of photosynthesis, but a result of the balance between photosynthesis and respiration. So now, let's dig into these 2 IB syllabus statements. State that oxygen is produced in photosynthesis from the photolysis of water. Energy is needed to produce carbohydrates and other carbon compounds from carbon dioxide. Photosynthesis broadly has 2 sets of reactions: the light-dependent reactions and the light-independent reactions. The light-dependent reactions require water, and light results in the photolysis of water, and oxygen is given off as a waste by that process. the energy provided by light is transformed into the chemical energy of ATP and NADPH. NADPH is a molecule like NADH, introduced to you when we studied respiration. The light-independent reactions take carbon dioxide along with the energy stored in ATP and NADPH to form the energy-containing carbon compounds such as glucose. Oxygen is produced in the light-dependent reactions with the photolysis of water, and the energy stored in ATP and NADPH from the light-dependent reactions is used to fix carbon dioxide into carbon compounds. Oxygen is produced from the light-dependent reactions when light energy lyses water. The light-dependent reactions produce ATP and NADPH as energy molecules available to fix carbon dioxide into organic carbon compounds such as glucose. Now, both sets of reactions, the light-dependent and light-independent reactions, occur within the chloroplast within a plant cell. Now, let's take a step back to look at how the leaf and the chloroplast are well suited to perform these reactions. Leaves take up carbon dioxide from the air, and water arrives to the leaf in the vein from soil water through the roots. Solar energy is absorbed by cells in the leaf, cells with chloroplasts. The chlorophyll within the chloroplast is the molecule that absorbs solar energy, specific wavelengths. The high surface area provided by the flat shape of the leaf facilitates the absorption of solar energy and the uptake of carbon dioxide. Here is a cross-section of a leaf. The entire middle section of the leaf is called mesophyll. Carbon dioxide enters through pores on the underside of the leaf. These pores are called stomata. The vein brings water to the leaf from soil water through the roots. The upper layer of the mesophyll, called palisade mesophyll, is a densely packed layer of cells, each of which has numerous chloroplasts. The chlorophyll is found within the chloroplast. Here is an image of the stomata, typically found on the underside of the leaf. Stomata are pores that allow carbon dioxide to diffuse into the leaf for photosynthesis. Open stomata also allow water to diffuse out in a process known as transpiration. Here are two closely related IB syllabus statements. The upper one is for SL and the lower one for HL. The two statements are nearly identical in their intention, even though the second one is for HL only. Explain the structure function of organelles within the palisade mesophyll cells of a leaf. Explain how the structure of a chloroplast is adapted to its function in photosynthesis. In this diagram of a cross-section of a leaf, the palisade mesophyll is a layer of cells near the top of the leaf, receiving the highest amount of solar radiation. The densely packed palisade layer has cells with numerous chloroplasts with which solar energy can be absorbed, specific wavelengths. Water arrives to the leaf in the vein, specifically the xylem vessels. Carbon dioxide enters the leaf through stomata. Remember, leaves are flat, and this flat shape provides a high surface area, and the high surface area serves for the absorption of solar energy and carbon dioxide. In this diagram of a leaf cross-section, we can see the palisade mesophyll as a layer of cells densely packed to maximize solar energy absorption. Water for photosynthesis arrives in the vein from the soil water through the roots, and carbon dioxide enters the leaf through stomata. Again, the thin shape provides for high surface area. High surface area serves the absorption of solar energy as well as carbon dioxide. Chloroplasts seen in this photo are small oval-shaped organelles that have a high surface area due to their small size and oval shape. Within the chloroplast are many membranes into which chlorophyll is embedded. Chlorophyll reflects green wavelengths as it absorbs blue, and red wavelengths. In this diagram of a chloroplast, you can see numerous flattened membranes into which chlorophyll is embedded. The numerous flattened membranes create a very high surface area for the absorption of solar energy. The light-dependent reactions happens on these membranes. The light-independent reactions occur in the fluid surrounding the membranes. As an aside, notice that the chloroplast is composed of 3 membranes, 2 outer membranes and the inner membranes known as thylakoids. More on thylakoids upcoming. The chloroplast has the double outer membranes here and numerous inner membranes throughout the chloroplast. These inner membranes create high surface area for the absorption of solar energy. The light-dependent reactions happen on these membranes known as thylakoid membranes. The light-independent reactions occur in the fluid surrounding the thylakoid membranes, and the fluid is called the stroma. Here are 2 IB syllabus statements that are appropriate right here. For SL students, there are a couple of syllabus statements still ahead that are required of you. So even though these statements are for HL students, don't turn the movie off just yet. Draw and label the structure of a chloroplast to indicate the adaptations of a chloroplast to its functions. Explain how the structure of a chloroplast is adapted to its function in photosynthesis. Here is a diagram of a chloroplast. You can see the double outer membranes. You can see the thylakoid membranes arranged in stacks. The lumen of the thylakoids plays an important role, which we will get to later for HL students only. The light-dependent reactions occurs on the thylakoid membrane, while the light-independent reactions occur in the stroma. In this micrograph of a chloroplast, you can see the region called the stroma, and you can see the thylakoid membranes. You can see that the thylakoids, if you look closely, have an interior compartment. It's called a lumen. As well, in this micrograph, we can see starch within the chloroplast. This should make sense to you. Starch is composed of glucose, and glucose is the principal organic product of photosynthesis. This is a diagram that I've shown a few times now. You can see the thylakoid discs arranged in stacks called a granum. Each stack is called a granum. Many stacks are seen here. Grana is the plural. You can imagine the interior compartment of the thylakoid called the lumen of the thylakoid. And chlorophyll is embedded on the thylakoid membrane, and this is where the light-dependent reactions occur. Here is our last image of a chloroplast. The thylakoid membranes are clear. The lumen of the thylakoid discs is clear to see. The region with fluid called stroma is also clearly labeled. The light-dependent reactions occur on the membranes of the thylakoids, while the light-independent reactions occur in the stroma. The lumen of the thylakoids has an important role to play. More later. Be patient. Work on vocabulary. The light-dependent reactions occur on the thylakoid membranes, and you already know that ATP and NADPH are products from the light-dependent reactions. So with this information and your study of the mitochondrion and respiration, you might have expected protein complexes, in fact electron transport systems, to be embedded on the thylakoid membranes, and they are, as seen in photosystems 1 and 2 in green and red. As well, in the same manner as ATPases in the mitochondrion, there are ATPases on the thylakoid membranes. Also, notice the inner compartment of the thylakoid. This is important as well. Stay tuned. This table displays the protein quantity as a percent of dry mass of various membranes. The high quantity of protein in the thylakoid membrane, comparable to that of the inner membrane of the mitochondrion, is a hint of the importance of the thylakoid membrane in photosynthesis. Electron transport complexes and ATPases are found in high frequency on the thylakoid membranes. Chlorophyll is embedded in the thylakoid membranes, and light bumps an electron from chlorophyll to electron carriers in the electron transport systems on the thylakoid membrane. The electron bumped off of chlorophyll is replaced from water when solar energy results in the splitting of water, the photolysis of water. Remember that photosynthesis consists of light-dependent reactions and light-independent reactions. The light-dependent reactions occur on the thylakoid membranes within the chloroplast. The light-dependent reactions require water and light. The light-independent reactions occur in the stroma of the chloroplast. The light-independent reactions require energy molecules in the form of ATP and NADPH. As well, the light-independent reactions require carbon dioxide. The ultimate product of photosynthesis is an organic carbon compound with chemical energy, glucose. And that brings us to our last IB syllabus statements for all students, SL and HL combined. Outline the effects of temperature, light intensity, and carbon dioxide concentration on the rate of photosynthesis. State that temperature, light intensity, and carbon dioxide concentration are limiting factors on the rate of photosynthesis. The two statements are clearly related. The chemical reactions that comprise photosynthesis involve enzymes. As such, the curve that displays the rate of photosynthetic reaction as a function of temperature follows the well-known enzyme curve. Reaction rate increases as temperature increases due to increased molecular motion, which increases collisions among reactants. But when the temperature reaches a high value, enzymes denature and the rate of the reaction declines dramatically. Take note, low temperatures can limit rates of photosynthesis, as can high temperatures. So let's move to light intensity. The rate of photosynthesis increases as light increases. More light stimulates greater production of ATP and NADPH, both of which serve to fix carbon dioxide into carbohydrates. But at a particular light intensity, the rate of photosynthesis reaches a maximum. There is a maximum amount of chlorophyll to absorb light. There is a maximum number of electron transport systems on the thylakoid membrane that produce ATP and NADPH. Thus, light levels higher than value D do not result in greater rates of photosynthesis. The rate of photosynthesis reaches a maximum value despite greater quantities of available light. This graph has similarities to the last graph as it displays rate of photosynthesis against available light on the x-axis. But this graph adds carbon dioxide as a third abiotic factor that influences the rate of photosynthesis. Compare the middle curve with the upper curve. The middle curve represents the photosynthetic rate at 25 degrees C with 0.04% carbon dioxide concentration in the air surrounding the plants. The upper curve represents the photosynthetic rate at 25 degrees C with a 0.4% concentration of carbon dioxide in the air surrounding the leaves of the plants used in this experiment. At a constant temperature, an increase in carbon dioxide results in higher rates of photosynthesis. The lower curve here, this curve, relative to the upper curve, suggests that carbon dioxide can be limiting. This curve displays the rate at 15 degrees C, and if we compare it to this curve at 25 degrees C, we can also note that temperature is a limiting factor. Here are three curves representing different abiotic conditions. Rate of photosynthesis is given on the y-axis and light intensity on the x-axis. Right away, we can say that low light conditions at, say, W limit photosynthesis, no matter what the temperature or carbon dioxide concentration. At a constant carbon dioxide concentration, the X curve represents a cold condition relative to the Z curve. If temperature becomes too high, rates of photosynthesis would decline dramatically due to denatured enzyme. At a constant temperature, the X condition represents a low carbon dioxide concentration relative to curve Z. And lastly, as we push to the final few slides of this movie, the photosynthetic pigments within a leaf can be separated based on their molecular size and their interaction with a solvent and with chromatography paper. A leaf suspension is applied to chromatography paper, then allowed to dry. The paper is dipped into a solvent that runs up the paper by capillary action. The pigments separate from one another based on their size and their attraction to the solvent or the paper or both. Here is a diagram of the pigment application, and then the chromatography paper is dipped into a solvent. The solvent runs up the paper by capillary action, separating the pigments. The separation of pigments depends on the molecular interaction of the pigment with the solvent and the paper. If the solvent is nonpolar, it would carry a nonpolar pigment further up the paper. In the photo on the left, you can see the application of leaf pigments onto the paper, and on the right, we can see the results of a chromatographic separation. RF values can be calculated for each pigment and compared to published values, assuming the same solvent and chromatography paper have been used. The RF value is calculated as distance traveled by the pigment in centimeters, divided by the distance traveled by the solvent front in centimeters. For example, here is the distance traveled by this pigment, and we would divide that by the distance in centimeters traveled by the solvent. RF values are expressed as numbers between 0 and 1. And that brings us to the end of IB Bio Photosynthesis Part II.